Cancer is one of the leading causes of death worldwide. To treat cancer, various approaches are applied, such as surgery, radiation, and chemotherapy. In efforts to reduce systemic toxicity of highly toxic agents which are used to kill cancer cells, targeting drug delivery systems are applied. Among them, the most popular are antibody drug conjugates, peptide drug conjugates, and nanoparticles charged with cytotoxic agents. And in recent years, albumin-based drug delivery systems attracted the attention of researchers. Human serum albumin is a very abundant protein in our body. It accounts up to 60% of all plasma protein. It is highly water-soluble, non-toxic, and possesses extremely long circulation time, up to 21 days. Here in this table, I tried to compare the most popular covalent drug delivery systems, ADCs and PDCs, with albumin drug conjugates, covalent albumin drug conjugates. And you can see that in many respects, albumin drug conjugates combine the advantages of both ADCs and PDCs. Like ADCs, albumin drug conjugates have long circulation time, several days. And like PDCs, they have a low toxicity, low risk of immunogenicity, and the ability to accumulate in tumor site. Here you can see some albumin-based drugs which are either in clinic or in clinical trials. And approximately half of them are designed to treat cancer. For example, nanoparticles Abraxane and Fiara have been approved for clinical use. Other compounds like MTX, HSA, and aldoxorubicin are in clinical trials. Why albumin as drug delivery system is so attractive for tumor targeting? Tumor grows very fast, and in order to provide it with nutrients and amino acids, vascular system should grow fast as well. And this fast growth doesn't allow a vascular system to mature. So vascular system of tumor is very immature and highly permeable even for macromolecules. At the same time, lymphatic drainage system is insufficient to effectively wash out all these macromolecules. And these two reasons contribute to the so-called enhanced permeability and retention effect, which allows macromolecules to accumulate in tumor tissue. Any compound before entering tumor cell has to overcome two barriers. First barrier is epithelium layer between the circulation system and tumor tissue. And the second barrier is cell membrane around the tumor cell. And for ADCs, it was shown that the first barrier is time limiting. It is much higher comparing with the second one. So the penetration from a blood vessel to tumor site proceeds in 100 times lower rate comparing with following receptor-mediated penetration inside tumor cells. And no wonder that less than 1% of antibody drug conjugates, as well as less than 1% of nanoparticles, can reach the tumor site. In order to overcome this disadvantage, some variants of antibodies have been suggested, like as mini-body or dia-body. Albumin is twice as small as ADCs. Molecular weight of average ADC is around 150 daltons. Molecular weight of albumin is 67 kilodalton. And it was shown for MTX-HSA, covalent conjugate of methotrexate and HSA, that 14% of this conjugate can penetrate into tumor. So albumin is much more permeable for epithelium layer separating blood vessel and tumor tissue. In addition, there are some active transport through membrane, through epithelium layer, such as GP60 receptors, which catch the albumin inside some vessels, and this vessel transcytoses into the tumor tissue. Accumulation of albumin in tumor tissue can be visualized using Evans blue. It is known that there are approximately 10 sites for Evans blue binding on the surface of albumin, and once injected into bloodstream of mice, you can see the distribution of albumin. And here you can see that albumin is accumulated in tumor tissue. The second advantage of albumin is its very long circulation time. You can see that circulation time of albumin-based conjugates is approximately the same or comparable with circulation time of ADCs. Why it happens? Because both of them, IgG, which is the base of most ADCs, and albumin can bind to FcRn receptors. These receptors, this binding is very pH dependent. So when albumin or IgG is captured to epithelium cells, it binds to FcRn receptors, but this binding is strong only in acid medium. So you can see that after binding of albumin to FcRn, all unbounded proteins are sorted out for degradation in lysosomes, and albumin or IgG is released back into circulation. So let's consider how it happens. Here you can see that there are two binding sites on FcRn receptors, one for IgG, another one for albumin. And IgG binds with FcRn in its Fc region, rich with histidine residues. Albumin binds on the other side of FcRn, and in this region, FcRn also is rich with histidine residues. Why histidine? Because it is known that histidine can be charged or uncharged depending on pH of the medium. So it gives the opportunity to the binding to be very pH specific. IgG and albumin bind strongly to FcRn in acid conditions, but this binding breaks down in neutral conditions. The question is, how can we use these properties to increase the half-life time of small molecules or peptide drugs? First and very obvious idea how to do it is just to link peptide to either albumin or Fc region of IgG. And it was realized in two GLP-1 analogs, albiglutide and dulaglutide. In albiglutide, GLP-1 is fused to albumin. In dulaglutide, it is fused to. You can see that in both cases, half-life time of albiglutide and dulaglutide have been increased up to 5 days. So let's consider other strategies used to increase half-life time of GLP-1. You can see that simple peptide modification can increase half-life time, but only to several hours. We have already considered how Fc fragment or albumin attachment works to increase half-life time, but let's see on liraglutide and semaglutide. In these compounds, GLP is attached to fatty acids. Why fatty acid? Let's recall that one of the main functions of HSA in our body is to transport fatty acid and some other lipophilic compounds throughout the body. And to do that, there are several binding sites on the surface of the albumin to which fatty acid can bind. And when fatty acid is attached to peptide and binds to, and this conjugate is binds to albumin, albumin rescue the whole compound, the whole molecule containing fatty acid and peptide from the degradation in lysosomes. But it appeared that not only fatty acid possess such properties. There are several other compounds such as truncated Evans blue, para-iodophenyl acetic phenylacetyl butyric acid, and ibuprofen, and these compounds being attached to peptide also not only increase half-life time of these compounds, but also increase their accumulation in tumor sites. Another way how can we use albumin can be illustrated by aldoxorubicin and some other compounds which are developed by SIDRx company. In aldoxorubicin, doxorubicin is attached to reactive maleimide group. And once this compound is injected into the bloodstream, immediately the reaction occurs between albumin and this reactive group. And as a result, a covalent conjugate of doxorubicin and albumin is formed. And in clinical trials, it was shown that half-life time of these compounds is up to 40 hours. One more way is albumin nanoparticles. Two albumin-based nanoparticles, Abraxane and Fiara, are already in clinics. And despite the success, we can state that not all advantages of albumin can be used in case of nanoparticles. First of all, the size of nanoparticles is quite big, and that's the reason that only 1% and even less of injected nanoparticles can reach the tumor site. And the second disadvantage is that during process of manufacturing of nanoparticles, albumin is partly denaturated, so its binding with FcRn receptor is impaired, and the half-life time is, though it is higher comparing with, for example, docetaxel, but at the same time it is much lower comparing with albumin itself. So for Abraxane, it's only 27 hours. And another way is a covalent attachment of active component to albumin. For example, it was done an example of this approach is MTX-HSA, a covalent albumin conjugate containing methotrexate and albumin. And it was shown that for conjugate containing 1-to-1 ratio of payload and albumin, the half-life time of this compound is approximately the same then similar to half-life time of pure albumin. So it is up to 18 days. And it was shown in rats in preclinical studies that accumulation of MTX-HSA in tumor sites is up to 14% during first 24 hours. So let's summarize how can we use albumin as drug delivery system. First of all, um, uh, it is fusion of peptide or some other compound with albumin, so it can be illustrated by albiglutide and MTX-HSA. Another way is to attach some compound with non-covalently binds to albumin, such as fatty acid or some other compounds. This approach can be illustrated by liraglutide and semaglutide. Next way is nanoparticles and covalent conjugates, which forms in vivo, such as in case of aldoxorubicin. Now let me show you how we use albumin-based drug delivery system in our company. So combining highly active cytotoxin and human serum albumin, we created albumin-based compound TR101. And using Evans blue, we confirmed that TR101 can effectively penetrate into and accumulate to tumor tissue, but not only to tumor tissue, but it can penetrate through cell membrane and accumulate in tumor cells. Here are results of cell proliferation inhibition assay, and you can see that TR101 is at least as active as small molecular payload. Among various types of cell lines, we choose aggressive lymphoma cell lines to perform next xenograft experiments. As a positive control, rituximab was chosen. Rituximab is a chimerical monoclonal antibody against CD20, and it is approved against lymphoma as a monotherapy and as a part of combinational therapy R-CHOP. In addition, we studied small molecular payload, and the dose for small molecule was chosen based on its very limited solubility and high toxicity. TR101 demonstrated significant tumor growth inhibition in all experiments, comparable or even exceeding the effect of rituximab. But it is very interesting to compare TR101 and a small molecule. So a small molecule accounts for 1% of the weight of TR101. So TR101 in high dose, 200 mg per kilogram, contains 2 mg per kilogram of small molecules. But it is obviously that we could not use small molecule in such a high concentration because of both solubility and toxicity reasons. But conjugation with albumin allowed us to increase the dose to do it more effective. And the second thing, TR101 in lower dose contains 0.5 mg per kilogram of small molecules. Small molecule itself was used in a dose 0.4 mg per kilogram. So these are very comparable doses, but you can see that effect is completely different. So TR101, even in low dose, is much more effective comparing with small molecule. We believe that this is because TR101 brings payload directly to tumor site. So as a summary, albumin can extend circulation time, increase water solubility, lowers toxicity, and enable to penetration to tumor tissue. And let me say several words about our company. TIRCAN was founded by colleagues who worked together at Tulane University in the United States, side by side and in um, a part of laboratory of Nobel Prize laureate Andrew Shelley and Professor David Coy. Both of them are well-known, worldly recognized experts in peptide field. And our innovative drug development technology platforms of our company includes peptide compounds, peptide drug conjugates, and protein drug conjugates, but we focus mainly on albumin but not antibodies. Here is our pipeline. You can see that our tier 101 is now in IND step, so we passed through pre-IND step and now we are preparing all our documents for IND application. Thank you for your attention.